Hi friends, welcome back to KPS Bio Tutorials. It is the fifth episode of the lecture series Immunology. I request to all of you listen to all the episodes from the introduction onwards. For that, I have made a playlist namely Immunology in my channel page. So you may visit my channel page and here you can see the playlist, and in the playlist there is Immunology. Here you can see various subjects. Here you may select Immunology, and here you can see all the previous episodes from the part 1 to part 4. So you must listen to all the previous episodes, then only you can understand the subject very well. I also request you to subscribe this channel if you are not yet subscribed, and also enable the bell button, then only you will get the notification when I upload the videos. Today I would like to talk about the secondary lymphoid tissues or organs. In the last class we have been discussing about the primary lymphoid organs, that is thymus and bone marrow, in which we studied that from the primary lymphoid tissues or organs, the lymphoid cells, that is mainly the B cells and T cells, B lymphocyte and T lymphocytes, were developed and matured from the primary lymphoid tissue. So the major function of the primary lymphoid tissue was to produce an immunocompetent lymphocyte. But today I would like to talk about the secondary lymphoid tissues or organs. So let us go to the topic. Welcome back to our today's topic, that is secondary lymphoid tissue or organs. In the last episode we have been talking about the primary lymphoid organs, in which we discussed about the thymus and bone marrow, where we studied that the lymphocyte B and T lymphocytes were developed and matured from the primary lymphoid organs. So the function of primary lymphoid organ was to produce an immunocompetent B and T lymphocytes, whereas the secondary lymphoid tissues or organs, they are meant to provide an environment for the interaction between antigen and these matured lymphocytes. So the secondary lymphoid tissues or organs trap antigen and provide site for lymphocyte to interact with that antigen. The lymph nodes, spleen and mucosa associated lymphoid tissues, MALT, such as gut associated lymphoid tissue or GALT, are the secondary lymphoid tissues or organs. First of all, we can discuss about lymph nodes. You might be knowing about our lymphatic system. This lymphatic system consists of many lymph vessels with a number of lymph nodes. And these lymph vessels, many lymph vessels may join at the lymph nodes. So lymph nodes are a junction point of the joining of many lymph vessels. You might know the function of our lymphatic system. The blood capillaries enter the tissues and become numerous branches, and within the tissues the blood plasma, a part of the blood plasma may enter the interstitium or interstitial space or intercellular space, and this plasma then form the interstitial fluid. So the function of the interstitial fluid is to provide nourishment to the cells in the tissues, and also it receives the waste materials produced by the tissues. And this interstitial fluid then collected back or collected by the lymphatic vessels. An interstitial fluid collected to the lymphatic vessel are called lymph. This lymph there is a possibility of having antigens, because when the tissue was infected with an antigen or a foreign pathogen, it may enter the lymph, and the pathogens entering the lymph may be trapped within these lymph nodes. So the lymph nodes are acting as a sieve to filter these antigens entering the lymph. So the major function of these lymph nodes is to trap the antigens from the tissues and to develop appropriate immune response, because these lymph nodes are containing numerous lymphoid cells or immune cells. With the help of that immune cells, it can destroy this antigen and to produce appropriate immune response. So in this way, these lymph nodes protect our tissues from the antigenic attacks. The lymph collected by these lymph nodes then go back into our systemic circulation. Many lymph vessel joins to form large lymph vessels. Then the large lymph vessels will be connected to the systemic blood circulatory system, and in this way the reabsorption of the interstitial fluid into our circulatory system is taking place. When we analyze the structure of the lymph nodes, they are encapsulated bean-shaped structures containing a reticular network packed with lymphocytes, macrophages, and dendritic cells. These lymph nodes are consisting of different types of cells, mainly lymphocytes, macrophages, and dendritic cells. They form a reticular network within the lymph nodes, and these lymph nodes are clustered at junctions of lymphatic vessel. I already told they are found at the junctions of lymphatic vessel. And these lymph nodes are the first organized lymphoid structure to encounter antigens entering the tissue spaces. This is the first part of our body which can trap or which encounter with the antigen entering the tissue space. And morphologically, lymph nodes can be divided into three roughly concentric regions, namely cortex, paracortex, and medulla. Lymph nodes are divided into three regions: cortex, paracortex, and medulla. Here you can see in detail about the structure of the lymph nodes. Lymph node is divided into three regions: cortex, paracortex, and medulla. The outer region is called cortex, the middle region is called paracortex, the innermost region is called medulla. The cortex contains lymphocytes, mostly of B cells, and macrophages and follicular dendritic cells. So these types of cells, B lymphocytes, macrophage, follicular dendritic cells, and they are found a follicle, namely primary follicle. These cells are present in the form of a follicle, which is called primary follicle. It is said to be primary follicle before the encounter with the antigen. A primary follicle is the follicle which is not encountered with the antigen, so but it is ready for encountering with an antigen. The primary follicle then becomes secondary follicle when it encounters with an antigen. The secondary follicle is larger than the primary follicle, and it is because it contains numerous lymphoid cells and also a center, namely germinal center, and its application will be studied later in detail. So since this cortex region is mostly consists of B cells, they are called thymus independent area. Thymus independent area because it is consisting mostly of B cells and not T cells. So it is said to be thymus independent area because T cells are produced from thymus, not the B cells. Secondly, the paracortex, it is populated largely by T lymphocytes. It is a middle region. It also contains many dendritic cells which may be migrated from the tissues to the lymph nodes. So T lymphocytes are the most populated cell in the paracortex. So this area is said to be thymus dependent area. So this region, at the middle region, there is, you can see at this middle region is said to be thymus dependent area. It is consisting of mostly T lymphocytes. Then the medulla, in the innermost one medulla, which is sparsely populated with lymphoid lineage cells. Of those present, many are plasma cells. This medulla is very sparsely populated with lymphocytes, but the cells present here is mostly plasma cells. Plasma cells are cells which can secrete antibodies. So this is about the anatomy. This is about the structure of lymph nodes. Here you can see the histochemical studies or histochemical view of lymph node with the cortex, paracortex and medulla. So it is the histochemical analysis of the lymph nodes. So we can conclude that the lymph nodes which protect our tissues by trapping the antigens entering the tissues. The antigens entering the tissues may enter the lymphatic vessels, and from the lymphatic vessels it is trapped within the lymph nodes, and thereby our tissues are protected from the antigens with the help of this lymph node. Now we can discuss about the second type of secondary lymphoid tissue, that is spleen. Spleen is a large ovoid secondary lymphoid organ situated high in the left abdominal cavity. In the left abdominal cavity near to stomach, you can see an ovoid structure, namely spleen. And you can see here this spleen has a splenic vein and artery this splenic artery give the blood to the spleen, and from the spleen the blood is collected by splenic vein. Spleen is specialized for filtering blood and trapping blood-borne antigens. Unlike the lymph nodes, spleen filter blood and it destroys the antigens present in the blood. So it is a sieve for filtering the blood. In the lymph nodes filter the antigens in the tissues, whereas the spleen filter the antigens present in the blood. So blood circulates through the spleen and get filtered within the spleen, and thereby it is cleared of the antigens. More recirculating lymphocytes pass daily through the spleen than through all the lymph nodes combined. More number of lymphocytes are entering the spleen every day when compared with all the lymph nodes. So a spleen is the most efficient antigen filter when compared with lymph nodes, because more number of lymphocytes are passing through this spleen. So when we look at the structure of the spleen, it is surrounded by a capsule that extends a number of projections, namely trabeculae, into the interior to form a compartment-like structure. Here you can see this is an encapsulated structure, and its capsule extends inwards to the interior of the spleen, and thereby it forms number of compartments. These extensions are called trabeculae. These compartments are further divided into two regions, a red pulp and white pulp. The red pulp consists of a network of sinusoids populated by macrophages, RBCs and few lymphocytes. This red pulp, it is red because it is red in color due to the presence of RBC, and it also contains many macrophage lymphocytes. And it is the site where old and defective red blood cells are destroyed and removed. This is the site of destruction and removal of RBC. The old RBC, when the RBC, the lifespan of RBC about 120 days. After that 120 days, RBC get destroyed within this red pulp of spleen. The next part is white pulp. This white pulp, which is surrounding the branches of splenic artery. I told you there is a artery, namely splenic artery, which enter the spleen, and its branches, it divides into number of branches within the spleen, and these branches are surrounded by a region, namely white pulp, and this region consists of number of T lymphocytes, mostly large number of lymphocytes, T lymphocytes, and this area is thereby called periarteriolar lymphoid sheaths. This white pulp region surrounding these splenic artery branches are called periarteriolar lymphoid sheath, or PALS. The blood-borne antigens and lymphocyte enter the spleen through the splenic artery, which empties into the marginal zone. Here you can see the artery entering the spleen, and it carry blood which may contain antigens. Also it contains many lymphocytes, and they enter the marginal zone. That means the zone between the red pulp and white pulp. At the margin of red pulp and white pulp, these lymphocytes and antigens are emptied. And if there is any antigen in this incoming blood, which is trapped by the dendritic cells present here. I told you here there is many macrophages or dendritic cells. These cells will trap that antigens and carry it to these PALS, periarteriolar lymphoid sheaths. Lymphocytes from the blood also will be there in this region, and thereby with the action of these dendritic cells and lymphocytes, the initial activation of B and T cells takes place here. These cells come in contact with the antigen, and these B and T lymphocytes, mostly the T lymphocytes, they encounter with the antigen, and thereby the B lymphocytes and T lymphocytes are get activated. The activation of lymphocytes are the first step in the production of an adaptive immunity or adaptive immune response. It is the first step in the development of an adaptive immune response, the activation of B and T cells. After activation, they undergo proliferation, then differentiation. So they are the different process in the adaptive immune response. They will be studied in detail in later chapters. So this development of a adaptive immune response start here when antigen enter the spleen and it encounter with the B and T cells at the PALS region of the spleen. So let us conclude like that. The lymph nodes which trap the antigens coming from the tissues, and it protect the tissues out of these antigens, whereas the spleen which trap the antigens in the blood, coming through the blood, and blood is get filtered and purified of these antigens within the spleen. In this way, these two secondary lymphoid organs protect our body from the antigens. I hope all of you understood this today's topic. If you like this video, if you understood well about this topic, you will like the videos and make your comments, and I will be coming back with the remaining part of this topic in coming episodes. Till then, goodbye to all of you.